Okay, so welcome to Mitosis and Meiosis. So we're going to move from the cellular metabolism of what we find in the mitochondria and in the chloroplast, and now we're going to talk about what's going on in the DNA, what's going on in the nucleus, and the whole cell as it divides. There are two different ways it can divide. It can divide by mitosis, where it makes two exact copies, or you can divide by meiosis, where you make individual recombinations of genes from the parents. Okay, so bacteria go through a process similar to mitosis. It's called binary fission. What happens is, if you remember, bacteria have a singular circular DNA molecule, and it needs to be replicated or reproduced into two. Okay, so you have this site of origin in green here. This is where it starts. It goes in both directions until you have a complete copy of two. Where it ends is called the site of termination. Those two chromosomes go to opposite ends of the cell, and then you have a septum formed through the process of septation, and then that increases separation until you get two complete cells. Now, in eukaryotic organisms, chromosomes are linear, more complicated, and the process is more complicated. Every species has a different number and arrangement of chromosomes. So, for example, chimpanzees and humans both have 46 chromosomes, but the chimpanzees have slightly different structural components within their chromosomes. Additional or missing chromosomes are usually fatal, so there are some processes which lead to that. Chromosomes are composed of chromatin. So chromatin is DNA and the protein which is bound to it. Not all DNA is used evenly. You have heterochromatin DNA, which is just the stuff that's not transcribed into RNA or proteins. And then you have euchromatin, which is the stuff that is actually used to make proteins and the functional parts of the cell. DNA is one long single strand of chromosomes are one long single strand of DNA, which, if you uncoiled them, would stretch from here to the moon, because they are very long, 140 million nucleotides long in a typical human chromosome. Now, the DNA and the protein, in a structural level, at the very microscopic level, you have histones, which are proteins surrounded by DNA. Histones are positively charged, and DNA is negatively charged, and so they will form bonds. When you have a histone and DNA together, they form what's called a nucleosome. Nucleosomes can pack tighter and tighter together to make different structures, including what's called a solenoid, and you can see that here. This allows maximum compaction. We don't have enough room in our cells to hold all the uncoiled DNA. Now, an array of chromosomes is called a karyotype, and this is usually displayed visually. So here's a karyotype of human chromosomes. Humans are diploid, meaning they have two copies. The N represents to the number of copies. You can see we have two of each of these chromosomes. These chromosomes code for the same things, and we have 46 total. Haploid then would be one set of chromosomes, or one N. N refers to, again, the set of chromosomes. And if a haploid human cell would have 23 chromosomes. Pairs of chromosomes are called homologous. So these are two homologous chromosomes. This one is a homologue of this one. Now, during replication, you start out with chromosomes that just have one copy of all the genes. One single DNA molecule. After replication, you have chromosomes that have two copies, and each of those copies is called a chromatid. And each of those chromatids are held together by cohesion proteins. And each of those chromatids also have an area for attachment to microtubules called a kinetochore. So these are these are homologous chromosomes. Each homologous chromosome, so this would be one from the mother, one from the father. Each homologous chromosome has two chromatids that code for the same things and are exactly the same. Now, in this form, the chromosome is called a tetrad. So the eukaryotic cell cycle includes interphase and the M phase or mitotic phase. During interphase, this is where you have the functional part of the cell. Gap phase 1 of interphase is where you have cellular growth, cellular function. S phase is where you replicate the DNA. G2 phase is where you're going to prepare for mitotic division. So you're going to increase the number of organelles, increase in size. And then the M phase is where you go through the splitting of the chromosome and the cell, and cytokinesis then is the final part where you're splitting off. So again, the 3 phases of interphase, and each chromosome has a constricted point called the centromere. And again, that's where the kinetochore is, and each sister chromatid has its own centromere, and then when they are combined, they also have a centromere with cohesion proteins connecting them. So, mitotic phase is split into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and get into that. They start creating solenoids, and the histones pack tighter and tighter together. And then you have a spindle apparatus forming, which is these microtubules and the centrioles, and they're going to go to opposite ends of the poles and attach them to the chromosomes. The nuclear envelope is the main event in prophase which occurs. It starts to break down. In prometaphase, that's where there is no more nuclear envelope. The nucleus is gone. Now you have chromosomes floating freely in the cell. And then you have the microtubules attaching to each side of the chromosome, one to each kinetochore, and they start to congress. They start to go through congression and aggregate towards the middle of the cell. Metaphase, they're all lined up in the middle in an imaginary line called the metaphase plate. And this will eventually become the point of cytokinesis, or the point at which the cell will divide into 2 different cells. Anaphase begins when the centromeres of the chromosomes of the sister chromatids split, so the cohesion proteins are broken. The sister chromatids are pulled to the opposite poles, and there are 2 forms of movements then. Anaphase A, kinetochores pull to the opposite poles. Anaphase B, the poles move apart. So this part of the cell is moving that way, this part of the cell is moving that way. At the same time, these chromosomes are being pulled by these microtubules. During telophase, the spindle apparatus disassembles, so lets go of all these chromosomes, and you get the reformation of the nuclear envelopes. And now you have chromosomes without sister chromatids. Chromosomes begin to uncoil, and then you start to use them, and nucleolus starts to reform. Final stage is cytokinesis, where you have the splitting of the cell into 2 complete cells. Animal cells have this structure called a cleavage furrow. Plant cells have this structure called the cell plate, because they have a cell wall which needs to form as well. Fungi and some protists, the nuclear membrane does not dissolve. Instead, mitosis occurs within the nucleus, and then it splits with cytokinesis. Alright, so now we're going to talk about meiosis, but first we need to talk about the sexual life cycle. Meiosis is the process of making gametes, and fertilization is the process of those gametes coming together to make a diploid organism. Somatic cells, so these are your non-reproductive cells, have 2 sets of chromosomes. They are diploid. Your gametes are sperm and egg, have only 1 set of chromosomes. They are haploid. Offspring inherit genetic material from 2 parents, 1 egg, which is haploid, and 1 sperm, which is haploid, to make a diploid. So life cycles can vary from one organism to the other, but they have this alternation of haploid and diploid stages. In most animals, the diploid stage is the dominant, including humans, or that's where you have your multicellular mitosis occurring. And then meiosis occurs, and you have a haploid stage, but it's very short. Alright, so the formation of gametes is called, happens through the process of meiosis, which is also called reduction division, because you're dividing the cells and reducing the number of copies of genetic material. It's similar to mitosis, but it has two rounds of division, meiosis 1 and meiosis 2, each with their own prophase, metaphase, anaphase, and telophase stages. There's no DNA replication between meiosis 1 and meiosis 2, so there's no doubling of chromosomes between that. And the second meiotic division does not reduce the number of chromosomes, but it does reduce the number of copies. It separates the sister chromatids for each homologue. Alright, so we'll go through these after we talk about crossing over. So in meiosis 1 and anaphase to in prophase to anaphase, till they separate, these chromosomes will aggregate together and switch over certain parts of their chromosomes. The point at which that crossover occurs is called the chiasmata, and this contact is maintained until they split at anaphase 1. So in prophase 1, the chromosomes coil together, similar to prophase in mitosis. The nuclear envelope disappears, the spindle apparatus forms, each chromosome has two sister chromatids, and synapsis also occurs. So sister or homologous chromosomes will basically connect together, and that allows them to go through the process of crossing over. And they remain attached at this site called the chiasmata. Metaphase, they all line up in the middle. In metaphase 1, however, they line up side by side. Okay, so the chromosome from the mother and the chromosome from the father are right next to each other. Crossing over events occur. They also assort independently, meaning not all the mother ones are on one side and father ones on the other. They can have different variations and mix up between the two. It is random. During anaphase, the chiasmata break, the microtubules are attached, and then they pull them to opposite ends. Each pole now has a complete haploid set of chromosomes consisting of a tetrad each. During telophase, the nuclear envelope reassembles, and then cytokinesis allows the two cells to split. Alright, meiosis 2 then occurs after an interval of length, but it's pretty much the same. The only difference is in metaphase, rather than lining up side by side, they can't do that because they only have one copy, but they line up on top of each other and then they split, similar to mitosis. The final result is 4 haploid cells. So you had tetrads, which split in meiosis 1, go to opposite poles, and then those tetrads split individually into sister chromatids to make haploids. In animals, these then directly develop into gametes, the egg and the sperm. Plants, fungi, and many protists, they can actually make multicellular structures out of the haploids. Some errors will occur, and this one of the error which is where chromosomes do not separate correctly is called non-disjunction. So this here is a cell that has non-disjunction event occurring. Rather than 2 chromosomes going one side and 2 on the other, 3 went to one side and 1 went to the other. So this is called an aneuploid gamete. This one has 1 less than it's supposed to, and these 2 have 1 more than they're supposed to. And this is the most common cause of spontaneous abortion in humans, but can also lead to what's called Down syndrome. So meiosis and mitosis are different in 4 different ways. Synapsis and crossing over occurs in meiosis, not mitosis. Sister chromatids remain joined at their centromeres through meiosis 1. Kinetochores of sister chromatids attach to the same pole in meiosis 1. And DNA replication is suppressed between meiosis 1 and meiosis 2. Alright, here's the figure. We'll probably go over this in class for meiosis 1. Here it is for meiosis 2. And that's it.